Hello guys, welcome to Dr. Science. So today we are going to talk about the second part of your porphyrias. In yesterday's topic I already told in my first part, I already talked about the first half of the cycle, and in this part I am going to talk about the second part of the cycle, which is your porphyrin ring synthesis. So first I will talk with the basics, then we will go with the images above. So I already talked in my first video that we have now synthesized a molecule called as porphobilinogen. Okay, now this porphobilinogen, it is a imperfect precursor of your pyrrole rings. So basically, in order to form a protoporphyrin ring, suppose this is a protoporphyrin ring, we need to have a perfect pyrrole, 4 pyrrole rings. Okay, so these are your 4 pyrrole rings, and these 4 pyrrole rings should be in a perfect condition, right? Then only we can make a protoporphyrin ring. And to this protoporphyrin ring, if we add iron inside it, then entire compound is called as your heme. And if a globin chain is added to it, the entire compound is called as your hemoglobin. Okay, so we need 4 perfect pyrrole rings. So I already told you that the porphobilinogen ring, it is a imperfect precursor of pyrrole rings. So basically, suppose these are the pyrrole rings, they are randomly arranged and they are imperfect. As you can see, they are randomly arranged and they are imperfect. So what we have to do, we have to convert this porphobilinogen into a compound called as hydroxy methyl bilane. Now, why we are converting this porphobilinogen into hydroxymethyl Because hydroxymethyl it is a stable and a perfect, not a perfect molecule, it is a stable molecule. As you can see here, the pyrrole rings are arranged here in a linear fashion, right? The pyrrole rings are present in a linear fashion. It means the pyrrole rings are getting stitched here to this. Okay, they are getting stitched and they are in a linear fashion. Hence there are 4 pyrrole rings here. These are called as linear. Okay, let me clear this one. And this step is called as your linearization. Why linearization? Because porphobilinogen, they are randomly arranged, whereas in the case of hydroxymethylbilane, they are arranged in a linear fashion. Hence they are called as your linearization of pyrrole rings. The step is called as your linearization of pyrrole rings. To be more specific, we call this as linearization of tetrapyrrole rings. Since there are 4 pyrrole rings, it is called as your linearization of tetrapyrrole rings. So once again, I am going to tell that one. So porphobilinogen, which is an imperfect molecule and which are randomly arranged, are going to get converted into hydroxy methyl bilane. Now hydroxymethyl bilane, so I will draw this one here. So porphobilinogen are imperfect and they are randomly arranged, whereas in the case of hydroxymethylbilane, they are perfectly arranged, or we can say they are in a linear fashion. Okay, and the step here is called as your linearization of tetrapyrrole rings. Now what is the enzyme responsible for this conversion of porphobilinogen into hydroxymethylbilane? and not only that, here water is going to get added, hence the name hydroxy methyl bilane. What is the enzyme responsible for converting porphobilinogen into hydroxymethyl bilane? The answer is very simple. It is hydroxymethyl bilane synthase 1. Okay. Or simply we will call this enzyme as uroporphyrinogen synthase 1. Okay. So now we have formed a compound called as hydroxymethylbilane, but we know that in order to make a porphyrin ring, the pyrrole rings should be arranged in a cyclic manner, right? Like this. So obviously we have to make this linear tetrapyrrole rings into a cyclic manner. So the next step is going to make this linear polymer into a cyclic manner, and this step is called as your cyclization of tetrapyrrole rings. Okay, cyclization of tetrapyrrole rings, and the newly formed compound here is your uroporphyrinogen type 3. So uroporphyrinogen, it is a more refined what we say, it is a more refined product of your pyrrole rings, which is your uroporphyrinogen 3. Now what is the enzyme responsible here? The enzyme responsible here is your uroporphyrinogen, wait a second, uroporphyrinogen synthase type 3. Okay. So hydroxymethyl bilane, which is a linear polymer, it is going to convert it into a cyclic polymer, which is your uroporphyrinogen type 3. Now this, why the name uroporphyrinogen? Because these can be excreted in the form of urine. Now this uroporphyrinogen, again it is not so perfect. It has to make a complete protoporphyrin ring. So there are going to be a lot of additions in this thing. So uroporphyrinogen, it is going to get converted into a compound called as coproporphyrinogen. Now this coproporphyrinogen, copro means they can be excreted via your feces. Now this during this process, carbon dioxide is going to get kicked out from uroporphyrinogen 3. So the enzyme is your decarbox, hence there is a decarboxylation. The enzyme here is a decarboxylase. To be more specific, the enzyme is uroporphyrinogen decarboxylase, which will convert the uroporphyrinogen 3 into coproporphyrinogen 3. Okay. Now I would like to say something here. Okay, let me complete this one. So I will tell one of the clinical importance of this one. So once the coproporphyrinogen is formed, we are going to make some modification. This coproporphyrinogen is going to get converted into a compound called as protoporphyrinogen type 3, and from protoporphyrinogen type 3 we are going to form protoporphyrin ring. Now this protoporphyrin ring, it is a perfect form. So hence to this protoporphyrin ring, suppose here is a protoporphyrin ring to which pyrroles are attached. To this protoporphyrin ring, if we add iron, the newly formed compound is your heme. So suppose this is your protoporphyrin ring again with 4 pyrrole rings. Now we are going to add iron in the middle of this. So the newly formed compound is your heme. So what is the enzyme here? The enzyme which is going to add iron to this protoporphyrin ring is your ferrochelatase. Okay, ferrochelatase. Now this is one of the important enzyme here. So what are the two enzymes here in the above diagram? Okay, my iPad is kind of stuck. Please wait. So what are the enzymes which are converting the coproporphyrinogen into protoporphyrinogen? The answer is very simple. These are your oxidases. Here and here, it is a simple reaction. The enzymes are your oxidases. So finally, we have synthesized the entire protoporphyrin ring. So now I will discuss how we can stop this heme synthesis. So I already told you that glycine plus succinyl CoA eventually form a compound called as amino levulinate, okay, by the enzyme called as ALA synthase. And from ALA we are going to form the end product, which is your heme. Now this heme will react with a apopressor, okay. Now this heme and the apopressor can go and inhibit your ALA synthase. Hence this is called as your negative feedback mechanism. So if there is increased heme synthesis in your body, plus it will react with your apopressor. Now this apopressor can go and negatively inhibit the ALA synthase enzyme. Hence there will be decreased heme synthesis. Now hence the ALA synthase is called as your rate limiting enzyme. So that was about the heme synthesis. In my next video I'll be going to talk about different types of porphyrias, so please watch that one. So before ending the topic, I would like to show the cycle here, which is taken from your biochemistry textbook. So as you can see, the porphobilinogen, which is an imperfect and randomly arranged pyrrole rings, are getting converted into hydroxymethylbilane in the presence of UPS1 or hydroxymethylbilane synthase 1. And hydroxymethylbilane, it is a linear polymer of linear tetrapyrrole ring. Hence this step is called as your linearization of tetrapyrrole ring. And you don't have to you don't need this one. I will explain this one in next video, but take a look at here. So now the enzyme UPS3, uroporphyrinogen synthase 3, will convert the hydroxymethylbilane into a compound called as uroporphyrinogen 3. Now uroporphyrinogen 3 in the presence of decarboxylase uroporphyrinogen decarboxylase can get converted into coproporphyrinogen. Coproporphyrinogen can get converted into protoporphyrinogen and protoporphyrin by the enzyme oxidases. And finally the protoporphyrin ring, to this protoporphyrin ring we are going to add iron by the enzyme ferrochelatase, and the end product is heme. Now you are going to ask, sir, what is this uroporphyrinogen 3 is getting converted into uroporphyrin, and coproporphyrinogen is getting converted into coproporphyrin. I will talk about this one in my next video, because I want to keep this video as short as possible. And not only that, as you can see here, succinyl CoA and glycine and here is ala synthase, which is forming your amino level in it. Now eventually the end product is your heme. The heme will react with the upper pressure, and they both will go down like this, and they are going to inhibit the ala synthase enzyme by negative feedback mechanism. Hence this is ala synthase is your rate limiting step. So thank you for the video. I hope you understand this one. See you.